गुड मॉर्निंग एवरीबॉडी, वेलकम एवरीबॉडी। आज हम लोग जेनेटिक्स एंड एवोल्यूशन चैप्टर के ऊपर रिवीजन कर रहे हैं। ये चैप्टर बहुत ही इम्पोर्टेंट है क्योंकि इस चैप्टर के ऊपर लगभग 20 मार्क्स आएंगे। हालांकि इसमें 3 चैप्टर्स है, प्रिंसिपल ऑफ इनहेरिटेंस एंड वेरिएशन, मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस और एवोल्यूशन। अब ये 3 चैप्टर अगर ठीक से करेंगे तो इसमें 20 मार्क्स पक्का आ जाएगा, इसलिए इसके ऊपर कोई भी टॉपिक होगा तो हम रिवीजन करेंगे, ठीक है। तो अब आप देख सकते हो कि इस चैप्टर में आगे हमारा इंट्रोडक्शन दिया हुआ है। इंट्रोडक्शन में हमको वैटसन और क्रिक के बारे में थोड़ा जानना ज़रूरी है, ठीक है। वैटसन और क्रिक के बारे में थोड़ा जानना ज़रूरी है। उन्होंने ही ये क्या किया था, डबल हेलिक्स डीएनए का मॉडल एक्सप्लेन किया था और विल्किंस के साथ मिलकर उन लोगों को नोबेल प्राइज़ भी मिला था। इसके ऊपर हमने पहले भी डिस्कस किया है। आई हैव ऑलरेडी डिस्कस्ड अबाउट देम, वैटसन एंड क्रिक। सो दे हैव वर्कड ऑन DNA and then they made a model, that is a double helix model. So let's understand about the chapters now. Especially I am dealing with these 2 chapters. You can see principles of inheritance and variation. Over here we also know about Gregor John Mendel. He worked on principles of inheritance chapter. He is considered as the father of genetics. Gregor John Mendel को ही हम father of genetics कहते हैं क्योंकि उन्होंने पी प्लांट के ऊपर काम किया था और उन्होंने पी प्लांट का जो कॉन्ट्रास्टिंग कैरेक्टर है उनके ऊपर रिसर्च किया था और उनके मोनोहाइब्रिड क्रॉस, डायहाइब्रिड क्रॉस और ट्राइहाइब्रिड क्रॉस और बहुत सारे एक्सपेरिमेंट्स उन्होंने किए थे, इसलिए उनको क्या कहा जाता है, फादर ऑफ जेनेटिक्स कहा जाता है, ठीक है। सो अब आगे बढ़ते हैं इसमें। अभी आपको एक चीज़ याद रखना है, कोई भी क्वेश्चन हो ये टेक्स्ट बुक से बाहर नहीं आने वाला, इवन एक लाइन नहीं आने वाला, इसीलिए ठीक से आप समझिएगा। यहाँ पर कंट्रास्टिंग कैरेक्टर्स ऑफ पी प्लांट्स के बारे में एक्सप्लेन किया हुआ है। आपको याद रखने हैं 7 कंट्रास्टिंग कैरेक्टर्स है। वी हैव 7 कंट्रास्टिंग कैरेक्टर्स ऑफ पी प्लांट दैट वी शुड रिमेंबर. Okay, 7 contrasting characters. So what are the 7 contrasting characters that we should remember? They are yes, here it's given: seed color, flower color, pod shape, pod color, flower position and height of the pea plant. So these are the shape of the seed, color of the seed, color of the flower, pod shape, pod color and flower position and height of the stem. So it's very confusing. Yeah, that's why let's make some order. So what do you do is, first one you always keep height of the pea plant and the last one always keep the position of the flower, and then remaining you keep 3 colors and 2 shapes. So that would be easy for you. Okay, in an order you keep, like first one you keep as height of the pea plant, that is tall and dwarf, and then keep the position of the flower, that is axial and terminal, and then go for writing 3 colors and then 2 shape. So that's easy for remembering. Okay, so here we deal with this, and then here you can see, you can just rearrange this order so that it will be convenient for you. Fine. So if it is given on the earlier side, that they are called dominant, and the one which is given behind, they are called as recessive. If it is given front, then that they are called dominant, and if they are given behind, then they are called as recessive. So let's go with the crosses that Mendel studied. All the crosses: law of dominant, law of segregation and law of independent assortment. All 3 together is called as law of inheritance. So let's see law of dominant. लॉ ऑफ डोमिनेंट का मतलब क्या है देखते हैं, क्योंकि ये तीनों लॉ अगर पूछा गया कि लॉ ऑफ इनहेरिटेंस को एक्सप्लेन करो करके पूछा गया तो आपको तीनों लॉ एक्सप्लेन करने होंगे: लॉ ऑफ डोमिनेंट, लॉ ऑफ सेग्रीगेशन और लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट। इसको शॉर्टकट में हम बनाएंगे। जब हम लॉ ऑफ डोमिनेंट बोलेंगे तो सिंपली याद रखना, जब हम मोनोहाइब्रिड क्रॉस करते हैं तब एक हाइब्रिड में अगर हम देखेंगे, एक टॉल और डॉर्फ का एग्जांपल देते हुए हम अगर हाइब्रिड में देखेंगे तो एक कैरेक्टर को टॉल बोलेंगे, दूसरे को डॉर्फ बोलेंगे, और टॉल का कैपिटल टी के से इंडिकेट किया जाता है अपर केस से, और उसी के लोअर केस से रेसिसिव को इंडिकेट किया जाता है। जो भी कैरेक्टर डोमिनेंट कहा गया वो एक्सप्रेस करेगा, अपने आप को दिखाएगा, फिनोटाइपिकली दिखाएगा अथवा जेनेटिकली वो दिखाएगा, एक्सप्रेस करेगा। और जो भी रेसेसिव हमने कहा वो एक्सप्रेस नहीं करेगा हाइब्रिड में। सो यही हुआ हमारा लॉ ऑफ डोमिनेंट। सो एज पर लॉ ऑफ डोमिनेंट in a monohybrid cross, when you see in an hybrid, one of the factor is dominant and is expressed, whereas another factor is recessive and is not expressed. So it is written in dominant character is written in trait is written in capital letter, whereas recessive trait is written in lower letter. So that will be the definition of law of dominant. Along with this, two more points you can add. You can say Mendel called genes as factor, and factors always occur in pairs. So if you add this, your law of dominant is complete. Now let's go for law of segregation. Law of segregation, that's also in monohybrid cross. When we see the transfer of single character from parents to offspring, when we see the transfer of single characters from parents to offspring, that means monohybrid cross. In a monohybrid cross, while formation of gametes, one of the factor actually factors get separated, and one of the factor enter one gamete, another factor enters another gamete. मतलब ये भी हमारा लॉ ऑफ सेग्रीगेशन भी हमारा लॉ ऑफ सेग्रीगेशन भी हमारा जो है मोनोहाइब्रिड क्रॉस का ही एक पार्ट है, जिसमें बोला गया है कि जब गैमेट्स बनते हैं उस समय गैमेट्स जीन्स जो है एक साथ रहते थे वो अलग हो जाएंगे, जीन्स अलग हो जाएंगे, एक एक गैमेट्स के अंदर एक एक जीन्स चला जाएगा। इसी को हमने लॉ ऑफ सेग्रीगेशन कहा, मतलब सेग्रीगेट मतलब सेपरेट, ठीक है। सो व्हाट वी कॉल लॉ ऑफ सेग्रीगेशन मीन्स ट्रांसफर ऑफ गैमेट्स। एक्चुअली when you study the transfer of single character from parents to offspring, then you find that while formation of gametes, factors get segregated and one factor enter each gamete. Yes, that's called law of segregation. It's also called as law of purity of gametes. It's also called law of purity of gametes, remember. So what is law of purity of gametes? In that way twisted questions can come, so you have to manage it. So I hope I'm clear what I'm talking about. Now let's understand about other things here. The F1 generation. Let's see about monohybrid cross. You know how to do the monohybrid cross. You need to take to one tall and one dwarf, and you need to just cross them, and you see that in F1 generation you get both of them are tall. And then when you get both of them are tall, that means the ratio of phenotype character is 100%, whereas even the ratio of genotype character is also 100%. That's what we can tell in F1 generation. But when we see in F2 generation, we get 3:1, that is tall and off, whereas 1:2:1, that is capital T, capital T small t, and small t small t. That's what it means genotype. Okay, so here homozygous means we are using the same alleles, capital T or small t small t. If we are using different alleles, then that's called as hybrid or heterozygous. So phenotype means the expressed character, whereas genotype means it's a genetical combination or genetical makeup of a character. Genetical makeup of a character is called genotype, whereas phenotype means it's the expressed character. Okay, and that's called as phenotype. Now here dominant means the character that is expressed is called dominant and written in capital case, whereas recessive means the character that is written in lower case and is not expressed or hidden during hybrid, whereas it will be expressed when it is homozygous condition, that is small t okay. And whenever we do this cross, always represent the cross in a Punnett square. Punnett is the scientist name. He has given a square so that it is very convenient for the big flowcharts to be expressed, and in short it can be explained. So that's why Punnett square is very important. So you can draw the Punnett square. Okay, so and when we talk about dihybrid cross, we got the ratio, right? So now let's go with the dihybrid cross. When we talk about dihybrid cross, in dihybrid cross, we see the inheritance of two characters. Yes? At the same time, we see the inheritance of two characters. So that's what is called as dihybrid cross. And in monohybrid cross, we know that law of segregation is universal, whereas universal means, there is no modification, whereas law of dominant has some exceptional. And what are that exceptions in law of dominant? That is codominant and incomplete dominant. If they ask you the question, what are the exceptions of law of dominant, then you need to say it's incomplete dominance and codominance. Incomplete dominance means you have to give an example of Antirrhinum species. where you see that the ratio or in let's say in F2 generation is not the same as early, and even F1 generation ratio is not the same. So like when we are saying F1 generation, we get all if we are crossing white flowers with the red flowers, we get pink flowers in F1 generation. No flower is red, no flower is white. And then F2 generation, the ratio becomes 1:2:1 in both the case phenotype as well as genotype. That's what we see in case of incomplete dominance. So that you can see the crosses are same, you no need to change any cross, but only the values are different. So that's what is about one exception of monohybrid cross, that is law of dominant. Whereas another exception is codominant. We know in codominant we see in a blood grouping, you can see blood grouping alleles are written with the I letter. On the top we write A, B and O. That's why it makes easy. So A, B, and in O case we do write small I. So that's what it's represent, that it is a recessive. So here you see A is dominant, B is also dominant, whereas O is recessive. That's why A and B, whenever they come, they be together, and both of them are equally dominant, so equally expressed. So its value and ratio are similar to that of incomplete dominance. Only the thing is we need to know the example. In a blood grouping ABO, A, B are codominant. That's what we should remember. Okay, so that's what the exceptions of law of dominant. Let's talk about the dihybrid cross, where we see the inheritance of two genes. So during that time we have to go for the two characters. Any characters you can go, round and wrinkle, yellow color, any two characters of that 7 contrasting character you can choose and go for the cross. It's same like monohybrid cross, only the thing is you are mixing the two things. Like you see round and yellow, so round capital R, capital Y, capital Y. Similarly here you can see capital R, capital Y, they are small, then they are big. Weird, as if you are doing 2 times monohybrid cross and you are mixing them. Like gametes also, there you saw 1 and here there are 2, and then when you do this is also similar, it's also similar. Okay, but only the thing is ratio is 9:3:3:1. So you have to do that by putting the gamete this side as well as this side. In both the sides you are putting the gametes, one side in female gametes, one side male gametes. So in this case if you just fuse them, that comes over here like that. You are fusing them and bringing them over here, and then you are writing the answer, that is round yellow how many, then round green how many, wrinkled yellow how many, and wrinkled green how many. So how will you get this ratio? 9. You need to see capital R, capital Y means they are both are dominant. Obviously they have round and yellow. So like that you should know the characters at least. If you don't know the characters, it's difficult. First in the parents itself, if you know the characters what are they, then it is easy to do it over here. Okay, so this is about dihybrid cross. Okay, so similarly you have genotype ratio also, that is 1:2:1:2:1:2:4:1:2:1:1. So that means it's a genotype ratio. So that ratio you can just write 4 times 1, 4 times you can write 4 times 1, 4 times 2, and 1/4. That way also you can write the ratio. Okay, for the dihybrid cross. Now let's see chromosomal theory of inheritance. Chromosomal theory of inheritance was given by, yes, it was given by Sutton and Boveri. Yes, Sutton and Boveri given chromosomal theory of inheritance. In this theory, what did they say, Sutton and Boveri? Here they say that like genes, how they transfer from one generation to another, similarly chromosomes also do. And like genes occur in pair, here chromosome also occur in pair. Similarly, while segregation, they also get separated. Here also they get separated. So they compared the movements of the genes along with movements of the chromosomes. That's what it is. It is not so you can just check over here. And very importantly, this here you cannot differentiate between these two because both of them are nearly similar. Here also you can see. So now the question, Drosophila melanogaster, it is not a bee, it's a Drosophila. It's not a housefly. It's not a housefly. It's a okay, Drosophila melanogaster. Its lifespan is very short, let's say 2 weeks. So it is easy to study. Like pea plant has so many good characters because of which Mendel experiments got success. Similarly, Drosophila also has some good characters, like it has so many characters without blending, it moves to the next generation, which is because of which it's very easy to study. So it has a red eye, blue eye, male is small, female is big. There are so many contrasting characters because of which which Drosophila melanogaster is also widely studied by the scientists for their experiments. So you can study here about linkage and recombination. Linkage and recombination. What is linkage? All the genes in a chromosome are linked, so that's called linkage. If they are linked they are present near to each other, this is called complete linkage. And if they are present very far away in the same chromosome, it's called incomplete linkage. Similarly, recombination, it's just complementary to linkage. If linkage is high, recombination is less. If recombination is less, then linkage is high. Recombination means, the while separation of the chromosomes during meiosis division, what happens is, if the linkage is high, then very less amount of the DNA is recombined, whereas if the linkage is low, then the very high amount of DNA is recombined. That's why it's called, they are just like opposite. Okay, so linkage and recombination. Recombination means during cell division, that is meiosis cell division, the crossing over occurs. And here you can just see these crosses, and here be very careful with this diagram, because it's very, very important. And many a times they don't ask everything, they don't draw everything, but they will give one of these clips, and they'll compare it, and 2 times they ask about what is this positive means. They ask what is this positive means. The positive means it's actually wild type. Wild type means it can indicate something, and whatever it is indicate, it's dominant then. So that's what its wild type positive means. Now here you need to mainly see about linkage and recombination. You see that if they are very near, you see the WY plus and W are very near, so the piece of DNA that is getting separated is very less. Over here W and M are very far, and that's why the piece of DNA that is separated is very high. So that's what few things you need to note down over here. So, and if you note down that, that's okay. And let's come to the sex determination. Compulsory question from sex determination will come. We have so many types of sex determination in human being. It is XXXY. XX indicates mother and XY indicates father. So, and when there is a gamete separation, they get separated and then fuse. And while fusing, obviously if XX is God, then that's a female, and if XY, then that's male. Same thing with the birds, but only the thing is, your male are having X and female are having XY. But just for indication, we cannot indicate them with the Y. We use other words, or else we will be confused. So we can say ZZ and ZW in birds. Whereas Drosophila, we have XXXY. There are some insects where we see XXXO type of sex determination too. So that's normal in some insects. Okay, so that's what it is. And here about sex chromosome, you understand there are total 46 chromosome in a human being. That means it's not that it should be only 40, sometimes it will be more, sometimes it will be less, and that's what it causes some diseases. So normally we know when we say 46, then that 22 pairs are autosomes and remaining are sex chromosomes. That's what just now we know, XX and XY. Along with them only it is 46. So remaining are 22 pairs are are autosomes. 22 pairs are autosomes. That means you can understand how important the autosomes are, because autosomes are same in male also, female also. That's why it is not repeated everywhere, so don't get confused. There are autosomes. How many autosomes? There are 44 autosomes, whereas 2 are sex chromosome. That might be XX and XY. Total it should be 46 in a normal human being. But if there is a problem and if you have genetic disease, then that might be a different case. Okay, if the father is having both XX or XY, in that way, if it is having same, then it is called male homogamity, and if it is having different, then it's called male heterogamity. Similarly, if mother is having XX, then it is called as female homogamity. If it is having XY, then it is called heterogamity, female heterogamity. So you have to understand that. So here we understood the sex determination in human being, how it happens. You can show in a small flow chart. So mutation, sudden and random change in a gene that leads to genotype and phenotype changes. That's what is called mutation. It's a sudden change in a gene. So that can be due to mutagens. That might be chemical mutagens like cigarette, tobacco, or maybe intoxicants, drugs, etc. That might be the cause for the mutation, which causes cancer and many other complications suddenly. artificially, so in our manipulating our genes. That's what mutation means. And there is physical mutations like UV radiation, and there are some nitrogen mustard gases like chemical mutagens, and biological, some viruses are there which causes mutation, and that can cause cancer and many other problems. Okay, what are mutations? They are the sudden random change in genes. That might be deletion mutation, that might be insertion mutation, that might be duplication mutation, that might be whole chromosome set changing mutation, that is called as euploidy and aneuploidies. So it can be any kind of mutations. Okay, for example, we have a point mutation called as sickle cell anemia. Yes, sickle cell anemia, the person dies due to anemic, as well as the cells cannot get retention the oxygen due to its shape. That's why there is a lot of problem with the oxygen in sickle cell anemia patients, and that's due to point mutation. That is, at single point there is a change in nitrogen bases due to which the mutation is occurring. Okay, so let's go with the genetic disorders. We need to know some genetic disorders. We also need to know sex determination in honeybees. Okay, honeybees, what is crisscross inheritance, that might also come, so be careful. You just need to study from extra material if you have old textbook like this, or else in new textbook it's given. So you need to know about pleiotropy gene, you need to know about polygenic inheritance, pleiotropy gene in polygenic inheritance, and then sex determination in honeybee. These are the three topics that you need to go for revision. Okay, what is a pleiotropy gene? When we say pleiotropy gene in polygenic inheritance, these two things just opposite. Okay, if more than one characters are influenced by a single gene, then that's called as a pleiotropy gene. And if more than one genes are inherited affecting for a single character, then that's called as a polygenic inheritance. So they are the two different things. So you can give some examples and you can explain. So let's go with the other things now. Pedigree analysis, that's very important. They will give you they will give you these two flowcharts. Among them, one flowchart they will give you, and they will ask you to identify what is it. So autosomal dominant trait or autosomal recessive trait. So the one of them, first one is autosomal dominant and the second one is autosomal recessive trait. So if the same question comes, just you write autosomal dominant trait and also autosomal recessive trait. So if the question is complicated, they will ask you to find out the genotype. You need to write down the genotype. So when you write down the genotype, if it is a autosomal disease, then you can go for writing A, capital A, and capital A, small a. In that way you can write, or you can write same letters everywhere. You can write capital A. For mother also, for father also, you can write capital A. For disease, you can put 2 stars, 1 star for recessive and 2 star for dominant. So I mean to say, if it is 1 star and if you are showing the disease, then that's dominant, and if you are putting 1 star and not showing the disease, then that's recessive. So in that way you can indicate the parents for autosomal. And for sex linked, you know, it is XX, XY. So XX for female and XY for male. So in that way you can give the characteristics of the genes. They will ask what are the genes, how do you represent, with gene number. They'll give 1 to 3 and they will ask you to find the gene number. So that's what it is. Okay, hemophilia. It is, I think everybody knows about hemophilia. Hemophilia means it's a disease related to sex-linked recessive disease. You know that. Yes, hemophilic means, you know, Queen Victoria and her descendants, her sons, they had hemophilia because she was a carrier for this disease. Hemophilic disease. In this disease, what happens is the blood, the clotting does not happen. So, okay, it is affected. So there is no production of the thrombocytes or let's say fibrinogens, because of which there is no clotting of the blood. And in this disease, that's linked into the X chromosome. That's why she can only give to the both. She can give to male also, female also, but you know, in female there is one more extra X. That's why that can easily become carrier, but in male there is no chance to become a carrier because Y chromosome do not have a gene for that. So that's why males are mostly is seen, whereas females do not show this disease. When we say sickle cell anemia, I already talked about this. You can see the gene, it's called point mutation. You can see in place of a normal gene, you see in place of T, it's replaced by A, and that's why the complement became A. So thymine, in place of thymine, it was replaced by adenine, because of which it became GGAG, which should give glutamine, but it gave GTG, which gave GUG, which gave valine. So in place of glutamine, it became valine, because of which what happened is the disease has come up. So that means the cell shapes became sickle shape, which is very not good for the person to take the oxygen properly. So you can see one more, phenylketonuria. It's affecting chromosome number 11, and it's phenylketonuria is a really autosomal recessive, and it's very bad disease, because it causes the deposit of ketone bodies in the body, in urine and brain, and it causes mental retardation and other very bad problems. So that's not good. So that's why here this phenylketonuria can be identified by seeing the urine test or maybe blood test. So phenylketonuria, ketone bodies, that is ketone derivatives, are formed. Okay, it converts the amino acid phenyl into tyrosine. Actually, that does not happen when there is this disease. So chromosome number 11 is affected. So chromosomal disorders, I talked about aneuploidies. You know aneuploidies, there are either one or two chromosomes are more or less. So that's what we write 2n minus 1, 2n minus 1, 2n minus 2, 2n plus 1, 2n minus 2, like that. So if it is 2n minus 2, it's called nullisomy. If it is 2n minus 1, it's called as monosomy. If it is 2n plus 1, then trisomy. And if it is 2n plus 2, then it is tetrasomy. So somy, you need to add it. So that's what it is. Now when we talk about Down syndrome, it's example of trisomy, where we say 2n plus 1. So that is 21st chromosome is extra. That's what you should know in Down syndrome. Also you should know some of the characteristics, like there is a congenital heart disease, forehead is very small, it has many cysts in the palms, so there are so many neural problems, so that growth is not proper. This is all the problem, Down syndrome. So this is also called as Mongolian disease, because it looks like Mongolian. So here it's Klinefelter's syndrome. Here Klinefelter syndrome, when we are talking about Klinefelter syndrome, you can see that this genetic disease is also caused due to presence of additional X chromosome. That's why XXY chromosome, maybe more than one X also it might be. So that is XY is a normal male, but when there is addition of X chromosome, then that becomes 47 or maybe 48 or maybe more, and it is called Klinefelter syndrome. In such however, even though the person looks like male, but it has so many feminine characters with breast development called gynecomastia and other things. So Turner syndrome, this occurs in female, and female with XX actually the female don't have one X chromosome. That's why it's a 45 number in chromosome, and lacking X chromosome is leading to a lot of complications. She is very criminal minded, her neck is webbed neck, and she's stunted growth, not properly, and emotionally unstable. These all are the features of Turner syndrome. So there may be many more. So for today this chapter is done. Let's go with the other chapter.